things are going on in a coral. These are coral polyps under a confocal microscope with their natural fluorescence, and in red, symbiotic microalgae called zooxanthella. Corals associate with microalgae in the genus Symbiodinium, from which they obtain up to 90% of their energy, photoprotection from UV light, and their characteristic greenish-brown color. Other microbial partnerships with bacteria, archaea, fungi, or viruses are also essential, yet much less understood. Microalgae, microbes, and coral hosts establish a net of metabolic exchanges which ensure the health of the living coral. All these compartments, the coral host and its associated microbiome, form a functional biological system, which is called the holobiont. Most organisms are holobionts, like us humans, with our millions of symbiotic bacteria. The coral holobiont is a coordinated system until disturbance occurs, and the partnerships within the host and the microbiome get disrupted, and corals tend to lose the symbionts and their coloration. Corals are very sensitive to raises in the seawater temperature, which triggers symbiont dissociation.
Gas! 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 Gas!
Good symbiotic microbes get expelled. This is the so-called coral bleaching caused by thermal stress. The loss of beneficial symbionts entails the pigmentation, corals become pale, and the reefs in the decay, disease, and often death of the coral. Bleached corals become highly stressed, and this is concerning as climate is predicted to become warmer and bleaching events more frequent and severe. Unfortunately, the future of our reefs is already uncertain due to climate change. However, a sliver of hope comes from the fact that sometimes some corals are able to recover their microbes, and these new symbionts may be similar or may be more resistant, providing higher tolerance to future thermal bleaching. In fact, there are coral colonies that have become more adapted and resist high temperatures, maintaining their microbiome intact. This past summer, in the island of Oahu, Hawaii, the water temperatures raised up to 31 degrees, leaving behind a massive coral bleaching in most of the reefs of the island. Especially in Kaneohe Bay, this bleaching was devastating. From the Gates Lab at the Hawaii Institute of Marine Biology, located on Coconut Island at Kaneohe Bay, 3 researchers are addressing this bleaching event closely. Our lab, located literally on the coral reef, allows us easy access to monitor our corals in the field, which can then be brought to the lab for molecular and physiological analysis. Sensor loggers measure physical parameters on the reefs, and we have tagged pairs of bleached and resistant coral colonies during the bleaching to monitor their status. Small subsamples from these tagged colonies were taken from the climax of the bleaching and afterwards in different time points of the recovery. We have extracted the DNA from those samples, and now we need your help to cover the cost of new generation sequencing, an approach that used to be very expensive but year per year becomes more and more accessible thanks to technology. New generation sequencing is the only way we have to precisely characterize the microbiome in our corals. This is why your help is key in this project. We target to sequence 2 marker regions from our total DNA that act as barcodes and which will allow us to identify the microalgae and bacteria present in our samples. All this work will inform us on which microbes are involved in the bleaching, recovery, and the resistance of our corals, in order to create tools to better manage the future of our reefs here at Kaneohe Bay, but also will provide new understanding on how to conserve the threatened reefs around our planet. Thank you for your help. Let's keep the corals staying alive.